is going to be as quick as it can be, review on protein synthesis. There are two steps to make a protein: transcription and translation. Transcription takes the DNA in the nucleus and makes it into mRNA, which is messenger RNA. This happens in the nucleus. It includes an enzyme called RNA polymerase, which is going to break apart the hydrogen bonds holding the DNA bases together. And it does this at just a particular gene, whatever the needed gene is, at a place called a promoter. So the entire DNA strand does not get made into mRNA, just the needed gene. Transcription makes the mRNA, the messenger RNA, to take the code. So here we have the DNA. It's unwound in that gene area, and the RNA strand is being created for that particular area. Notice this other DNA is not needed, so it is not unwound. Translation is then taking that mRNA and making it into a protein. This happens at the ribosome. The mRNA brings the code to the ribosome, and then tRNA, transfer RNA, brings amino acids that correspond with the code on the mRNA. The mRNA is read in 3 bases together called a codon, and then tRNA has the opposite of those 3 letters called anticodons. It is the codons on the mRNA that code for the amino acids. So here you can see we have our mRNA down here at the bottom. We're at the ribosome. Here's our tRNA with our complementary bases. So you see AAA gives you UUU, and AAA codes for lysine that we can see on our chart that we can use. The newly formed proteins aren't done yet. They have to get shipped through the endoplasmic reticulum to the Golgi apparatus, where they will be repackaged and folded into the correct shape. Then they can leave the cell if necessary. Here's a picture showing this. So we have the proteins being made here on the rough ER. It gets shipped and packaged through the ER to the Golgi. This is the Golgi apparatus. It takes it, repackages it, sends it into this vesicle, which then gets released into the atmosphere through the process of exocytosis. So this all works together.